So I'm going to talk about primarily the Physiome project and computational physiology. And I thought I'd just, particularly with Mike's sort of background, given that this is the 10th anniversary of Combine, just talk a wee bit about the history of the Physiome project. And I think it's these two efforts really have been quite complementary. The SBML came out of the realization by the molecular biology, biochemistry community that there needed to be standards to handle complexity, whereas the Physiome project and CellML, FieldML came out of more the physiology community, again recognizing the need for standards. And whereas SBML was primarily bottom-up in a sense, from molecular up, the effort in the Physiome project was much more top-down. It was thinking about organ physiology, and then that needed to link downwards. So a lot of the effort came from thinking about the physiology and the physics associated with the physiology. So here's the rough history. In 1997, IUPS, which is the International Union of Physiological Sciences, set up the Physiome Committee, which they asked me to chair. And the reason they asked me to chair that was that we'd been for many years working on cardiac modeling and thinking about bringing engineering concepts into physiology through multiscale issues for the heart. And that committee I ran for many years. It's now being chaired by Andrew McCulloch at UCSD. And then out of that committee came the commitment to really develop these standards, CellML, which most people here would know about, and FieldML for the more spatial side of things. And it started really at the same time as SBML, and as Mike said, it was quite a while before the two communities actually realized that they were trying to do very similar things from a, albeit from a slightly different perspective. And then NIH got into the act in 2003 with the IMAG consortium, which I think has been a great stimulus in the US for getting a lot of groups together to think about standards, and involves all of those organizations in the US, NSF, etc., as well as NIH, and is still going with an annual meeting. Grace Peng is really the driver behind that. She started it, and it's still really at the helm of IMAG. STEP was the European strategy that was driven largely by Marco Visconti to bring similar efforts onto the European scene, and I was involved with Marco and a number of others in writing a strategy for the European Commission, which then got funded under the Framework 7. So STEP was actually under Framework 6, but the VPH, Virtual Physiological Human Network of Excellence, got going under Framework 7. And then other initiatives like the DD Moore one, the German Virtual Liver Network, and then following on from the end of the funding period for under Framework 7, the VPH Institute was founded. And then for us more recently, the driver or the funding to really keep the momentum going for the Physiome project has come from the NIH, particularly through this Common Fund Spark initiative, which I'll talk a little bit about, and then more recently David Nickerson from Auckland, Andre and Herb and a number of others are involved in leading an effort to bring modelling into biology through this new P41 centre. And then again a wee bit of history. So this was a photo probably taken around 2007, 2008, and identifies some of the key people. Ilias Ikiavadis on the left was the person in the European Commission who I think had the foresight to realize the importance of standards and really drove the use of the funding for standards through the European Commission. Marco Visconti was the powerhouse behind the development of the VPH in Europe. Dennis Noble has always been helping, particularly from the IUPS perspective, and Yoshi Karachi in Japan as well. And then all those people at the bottom. Jim Bassingthwaite was a key driver of the Physiome in the early days. Paul Nielsen in terms of developing standards, and David Nixon, Andre is there as well, right from the start. And a lot of these people are still, well, nearly all of these people are still involved quite closely. So what I want to talk about is a little bit about how we began. I mentioned that the cardiac modelling was really what got us involved initially in Auckland on the need for multiscale modelling standards. So I'll talk a wee bit about that. I'm going to talk primarily about infrastructure standards rather than the science, but it's all being driven by the needs of the science. And then I'll talk about the particular challenge, some of the challenges for the Physiome project. I think everyone here understands why we need standards, but I'll talk a little bit about our approach to developing those standards around SalamL and the Physiome model repository, and then the open core software that Alan Garney has led. And then a more recent initiative, which I'll talk, just got one slide on, but which others here, particularly Karen Lundegaard, she's going to talk about that at a poster on later in the week, this new journal. And then I just want to finish with some thoughts around not so much the standards, but more the mathematics and the physics-based frameworks that I think we need to be focusing on to support. Now that many of the standards are in place, I think we can perhaps shift the focus a little bit to, from the modelling perspective, to the development of robust underlying maths physics frameworks to support multiscale modelling. Okay, so how we began. So the original aim was to be able to use engineering technologies to model the heart, use finite element methods, and we developed high order. We tended to build our models from very high order cubic and meter basis function elements. And the reason for that was really just because with very limited compute power, we needed to find very efficient ways of being able to capture complex anatomy and then the physics on that anatomy. And then that got incorporated. Increasingly we pushed down towards molecular scale and hence SalamL and the link with SBML group, but also pushed to the clinical end with the use of imaging technologies where the models could be used in the clinic to help with the diagnosis or therapy for various diseases. So the heart physiome was what started it all, but the framework for the heart is really completely comparable for any organ or organ system. And it is this multi-scale idea that you've got to be able to capture the physics at the whole organ level, but also capture tissue structure, be able to image and capture in mathematical form the structure of tissue, shown here as collagen networks, because they're critical to the function of soft tissues in particular, well, all tissues. But of course, a lot of the real complexity occurs at that cellular scale, and we quickly realized that if you didn't have standards in place, it just became a mess at the cell level. And then various efforts to push down to the molecular scale. So a huge range of spatial scales and comparable temporal scales as well. And then just one example of how these models at the tissue level are being used. This is work of Vicky Wang from Auckland, who did her PhD on a sheep model of heart failure. So what we find when we look at the structure of cardiac tissue by using confocal imaging techniques, we find that there's an orthotropic structure to it. The fibers lie in a particular direction, the muscle fibers. They're bound quite tightly into planes that are relatively loosely coupled to each other, so the tissue can shear fairly easily. And the shearing properties of that tissue become critical for the function of the heart to achieve a high ejection fraction. So in disease states, what happens is that very often that tissue gets, the collagen distributions change, the sheets can no longer slide as easily, shear as easily, and diastolic heart failure is often associated with changing material properties in that collagen matrix, which if we have models that can understand the relationship between structure and function at that tissue level, then we can begin to understand how the changing tissue properties affect the function of the heart as a pumping, for pumping blood. And so now we can look at the tissue, identify the constitutive properties from these microscopic images, knowing, having done tests on isolated tissue samples, and then make predictions of how the tissue performs under various heart failure conditions. And those predictions are very, very good. You can accurately predict the overall compliance properties of the heart from a knowledge of that underlying tissue, observed tissue structure. But that structure changes right around the heart, so you've got to be able to deal with the spatial variation of that structure. And then just a comment on where some of this work at the whole organ level is going. A lot of the recent work over the last 2 years, Richard Christie, who's driven this, has been developing what is essentially a biological CAD, computer-aided design platform. So the idea is that instead of just generating large numbers of elements to capture the anatomy of organs based on medical images, for example, what he's been doing is to develop techniques of algorithmically generating the anatomy, the structure, from underlying Python scripts, which then allow us to control the details of that anatomy for different species, and then build up generic descriptions that can then be fitted to particular examples of a particular species, but the underlying generation of the topology of that organ is really handled via a Python, an algorithmic environment, which you can then easily manipulate to give you the features that are observed. So for example, if you compare human, pig and mouse, they look very similar once you've adjusted for scale, but in fact there's some key topological differences that become crucial when you're trying to really model the function or embed neural data, as I'll talk about in a minute, into this. So for example, the human heart has 4 pulmonary veins coming into the left atrium, whereas the mouse has 3, and the way those enter is rather different. They form an ostium before they enter into the left atrium, and then otherwise topologically they're rather similar, but every species particularly has a variation in the topology of those entering vessels. And these models now have a lot more anatomical detail than they did in the early days. So Richard has now added the ability to have the appendages involved as well for either electrical or mechanical computation. And then a lot of these high order element models, when they're used in terms of physics, solving the equations, often it's useful just to be able to refine those meshes and put them out as quadratic or trilinear, or depending on the particular software requirements for the basis functions for those elements. But these CAD models, we call them scaffolds, are defined in a way which allows them to be refined very smoothly. So just change the level of resolution and you can put out a mesh at a much higher level of resolution based on always going back to that common coordinate framework for the scaffold to start with. So with that background to the heart Physiome project, I just wanted to talk a little bit about some of the generic issues across all organs for the Physiome project generally. And the challenge is that we've got 50, 60 years of molecular biology, fabulous data sets of knowledge about how things work at this molecular scale. We try and assemble those into something of the order of 300 cell types, and then we try and bring those cell types into physiological function at the level of tissue. And for me, this level is the biggest challenge from an engineering perspective, because typically you only have 3 or 4 cell types, 3 to 5 cell types defining the function at that level, but the 3D structure and organization of those cells and their relationship to the surrounding extracellular matrix becomes critical in defining the function of the tissue. So whether it's an acinus in the lung or a lymph node or a nephron in the kidney, cardiac sheet, it's all at about the scale of around a couple of 100 microns in 2 dimensions, typically a millimeter in another dimension, but it's at the diffusion limit. So a lot of the challenge I think from a multi-scale modelling perspective now, from an engineering point of view anyway, is how to get much, much better at generating the physiological function at this tissue level based on these relatively small number of cell types, but where 3D organisation matters a great deal. And then once you have function properly validated at that tissue level, it's a reasonably straightforward process to scale up. I mean, if you look at a kidney in a human, there's, of the order of 10 million nephrons, sorry, a million nephrons. For a mouse, the nephron looks pretty much identical, but you only have 100,000 of them. For an elephant, you have 10 million of them, but they look pretty much the same. So once you've got the definition or the function, structure-function relationships defined for those tissue units for any species, you've really got it for all species, providing you can then deal with the scale-up problem that you need generations of blood vessels and so on to be able to perfuse the tissue at that level. But the only difference, there's no difference at that level between a mouse, a human and an elephant. The difference is the number of them that you have. So the scale-up problem is a relatively straightforward engineering problem, but there's still a lot to be done, I think, at the level of that defining those tissue units and then linking them down to all the complexity that exists at the molecular level. But a lot of that ignores one crucial thing that I think we're becoming in Auckland more and more conscious of, the need to figure out how to integrate these systems much better, because most diseases, most chronic diseases, are never a disease of one organ or one organ system. There's comorbidities. Any disease, heart failure, diabetes, asthma, whatever it is, will involve multiple effects on multiple organs, and we need to get much, much better, I think, from the perspective of an engineering community in being able to handle that complexity across these organ systems and understand how to link them into integrative whole body systems in order to apply them to disease. So I wanted to give an illustration of just one of these integrating systems, and the reason that this one has become important for us is that we're involved in an NIH-funded project, SPARK, which I'll talk about in a second, which involves the autonomic nervous system. But just as a reminder about a little bit of physiology here, there are essentially three motor systems in the body. The one on the left is the somatic, the voluntary system that's about neuromuscular coupling, but under voluntary control of your skeletal muscles. The one in the middle is the autonomic system, which has three parts to it. The sympathetic, that's the fight or flight response. The parasympathetic, which counters that, more under eating or relaxation conditions. And then the enteric system, which is a specialized one for the gut. And then the third system is the neuroendocrine, which is the pituitary and the hypothalamus, and the release of hormones from not only the pituitary but other organs as well, operating through the cardiovascular system, often in synchrony with the autonomic control. So the body is a highly integrated system. We need to begin to think about how we create the structures that can treat it as a highly integrated system. And the one I'm going to just talk a little bit about is the autonomic system. So NIH set up through the Common Fund, set up this project a few years ago called SPARK, standing for Stimulating Peripheral Activity to Relieve Conditions, which, and the motivation for it partly came because a number of medical device manufacturers were saying to the NIH, we can produce these stimulators that get embedded into the body to correct certain pathologies, but we don't understand enough about the side effects that those stimulators are having because we don't know enough about the autonomic nervous system, where the paths go. I mean, it's a little bit ironic that huge amounts of money have poured into computational neuroscience in terms of the brain, but it's only relatively recently that the need to do a similar effort on the autonomic system, which is what connects the brainstem to the body, has to be, the autonomic system has to be examined in the same way. And if you look at some of the physiological responses that are under the control of the autonomic nervous system, here's some of them. But, it's the things you're doing every day, all the time, are often under control of the autonomic system. And every organ in the body is completely innervated by sympathetic and parasympathetic or enteric nerves, which are being regulated by, at a whole lot of scales, which I'll talk about in a second, which are governing the switching on to a larger extent or switching off of the function of that organ, fine-tuning the function of that organ. So it becomes absolutely critical to understand the normal physiology of that autonomic control in order to understand a lot of pathologies associated with dysfunction in that control system. So what NIH did was to fund a whole lot of groups in the US who are, or mostly in the US, who are doing all the experimental work across all these organ systems. And then they funded some groups there's one group looking after the databasing, and then there's another group, which is the Auckland group, which is mapping all of this data into a common framework. And then there's a third group, which is looking at the computational infrastructure to make use of those maps. So in terms of the mapping, one of the key parts of this is just being able to properly define all the pathways and annotate all the connections between different cell tissue types around the body and the connection to the brainstem. So what we do, and this work is led by Bernard de Bono and involves a lot of the curation now is being done by a group at UCSD, Marianne Matoni and colleagues, to create the workflow for all the experimental data, making sure that it meets minimum information standards and is curated and annotated before it goes into the databases that we then call on to map it into models of the body. So that's an epianatomy map of, just one example, of the structures particularly around the superior cervical ganglion in the neck and the links to the brainstem. Then this is what we then do with it. We turn it into what we call a flat map, where we've got the brainstem at the top of the spinal cord, all the sympathetic pathways that pass out through the stellate ganglia, for example, and into the various structures around the carotid, and then for the structures controlling a lot of the function in the neck region, into various tissues within that upper part of the body, and then various afferent pathways, not so easy to see here, but if you zoom in, those blue ones are giving the afferents that talk back to the brainstem. And then there's a corresponding parasympathetic system as well. So what we're doing is via that workflow through epianatomy and then into these flat maps, trying to define all the neural pathways for all the organ systems of the body, and then allowing users to dive into these maps, zoom in, and then click on points to retrieve data associated with the various parts of the autonomic system. And all this is getting launched in a week's time at a meeting in Los Angeles. The ISAN meeting is the International Neuroscience meeting, which is where all of this is kicking off shortly. So here's an example of how we deal with the data for the stomach, and this is work in Auckland by Kumar. So this is working with a group at Purdue led by Terry Powley, and what they do is do micro CT of, in this case, rat stomach. Kumar then fits the micro CT data to a scaffold, and then out of that extracts an average scaffold, and then that average can be used to bring in other forms of data at different levels. You typically have a submucosal plexus of neurons and then a myenteric plexus, so you've got to be able to define muscle layers and then the neural layers between those muscles in order to connect to the pathways coming from the autonomic system or the afferents. And then they measure individual neuron data by cutting the stomach and opening it out flat, which we then also do with the model so that we can treat the experimental data with that template fitted to that particular rat stomach specimen where they've imaged the neurons, and then having embedded those, mapped those neurons into as material coordinates into that scaffold, we can fold it back again into the 3D and bring in lots of data from different rats back into the one average scaffold. In the case of the heart, this just shows some of the neurons in the surface subepicardial region of the heart. So the heart is full of neurons, and that becomes critical to the fine-tuning of the function of those myocytes. And then the other thing that these scaffolds allow us to do is to compare across species. So if you look at a human colon, it has these haustra structures shown here, these bulges, and then it has things called tenai coli, 3 of them, which are like longitudinal strengthening lines, collagen and muscle, smooth muscle, that gives it more strength longitudinally, and then the propagating waves move down these. But you've got to be able to capture the details of the anatomy, both the centre line and the haustra structure, and the tenai coli. But for a pig, it's an extremely different structure to the colon, and very different structure to the haustra and the tenai coli as well. It only has 2 of these tenai coli. The mouse has none, and it has hardly any evidence of the haustra and a very different centre line. So with the scaffold approach, we can generate these alternative species models and then use that to help compare experimental data between species and then relate it back to the human. But the purpose of doing all of this is to model it and to understand the control that is going on all the time. So for the heart, down here are the beta and muscarinic receptors through the standard signaling, protein kinase A signaling pathways. But these are being regulated by sympathetic neurons that sometimes operate directly from, this is the stellate ganglia, sometimes they then pass sympathetic neurons into the ganglia that sit at the base of the heart, the neural plexus that sits at the base of the heart, which then innervate the adrenergic receptors and talk to the afferent receptors going back or to the parasympathetic system. So there's a level of control here, there's a level of control here, there's a level of control at the level of the stellate where you're also talking to other organs. There's a strong coupling between heart and lungs, for example, and then likewise control at the level of the medulla in the brainstem. So we have to be able to, as well as mapping these pathways, we need to be able to deal with the hierarchy of mathematic control algorithms, because this is crucial to normal physiological function and to pathology. A lot of pathologies of the heart are associated with dysfunction of autonomic control. And the way that some of the medical device companies want to help deal with that is by having stimulators that operate on the vagus or the sympathetic neurons. But you've got to know what all the other connections are to those neurons if you're going to not have disastrous side effects. Okay, so now back to the standards and working downwards to the more cellular and subcellular level. I think everyone here understands the problem of why we need standards and modularity. And this is really the framework that we've set up over a long period. David Nickerson, leads all the interaction, of course, with the Combine community, with CellML, SPML, ZML in particular, but increasingly I think NeuroML is part of our link into the neuroscience community. And then Alan Garnier has developed open core software, which is the primary CellML environment for authoring model and running models. And then the model repository, Physio model repository, which is run by Tommy Yu, is where we store the models and provide the workspaces for people to develop models before they're made public. And people here probably know already a lot about this, but the CellML effort has been going on for a very long time, since the late 90s, and I think like SBML, has reached a level of maturity. There's still plenty to do, but it's probably a lot of the effort now is going to be more, I think, on things like making use of ZML and the way that you bring in other standards around these models. Physio Model Repository has something close to 1,000 models, like biomodels. It's a large database of models that are encoded in these markup languages. These comments are ones from some time ago, but I think they nicely illustrate why it is so important to have to try and get the journals to adopt modeling standards, because we find in the SBML community with the biomodels database find as well that very, very few models are published in a way that other people can use them. It's just inevitable that you make mistakes, typographical or other mistakes, but our experience in terms of trying to get these models into the standardized form and dealing with the authors in that process is that there's virtually no one who doesn't want their model to be available to others in a curated form. So there's plenty of goodwill, I think, in the community, providing we can find the right mechanisms for giving people carrots as well as sticks to get the models into the standardized form. But it is, I think, pretty well accepted by the community that just publishing a model it might get a publication on your CV, but it's not going to help the community unless you put this extra effort in to make it useful for other people. So here's an example, just one example of the use of the SalamL framework for dealing with models at this scale. Nima is a student at the ABI in Auckland who's just finished his PhD on modelling epithelial transport in the gut and glucose uptake in particular. And so what he did was to model, using primarily literature data, model the properties of the various receptors, transporters involved in glucose transport, and then modelling each of those, checking that the models are agreed with the experimental data for each individual component, but then using the import facility in SalamL to be able to then form composite models that preserve a modular structure, which is so important, I think, to being able to build up these complex models. So I just want to I've got one slide on the Physiome journal, which in fact Karen is going to talk a lot more and talk to her at her poster about this, and she's got a lot more detail. But we realized the need for this a number of years ago when people are happy to work, to get help to turn their models into properly curated, annotated, standards-based models on a website like the Physio Model Repository or Biomodels. But there's very little incentive for people to do that voluntarily. It takes a lot more work to get to make your model available in that form for others. It's a lot easier, and we're guilty of this as much as anyone, it's a lot easier just to publish the model and move on and even trying to put your model available as source code on a website doesn't work. I mean, so often we find that the websites are not in sync with what's been published. The equations don't agree with what's published in what's in the figure in the publications. So we really badly need to have better incentives for people to put the effort in to making their models available in properly curated form, standard form. And so we thought that maybe the best way to do this is to set up a journal where you can only submit your paper to this journal where you're dealing with the documentation and curation and annotation if you've also got that model and the experimental data behind it and the whole reason for developing the model submitted and accepted for publication in a primary journal. So the Physiome is intended as a secondary journal, but it's intended to give a citation credit for people that want to put in this extra effort, and it will always make reference back to the primary journal, because we absolutely don't want to compete with the primary journal, but it means that you're getting a second publication, and it could be a different author. It could be the person that's put all the effort into developing the model becomes the first author on the Physiome paper, whereas it may be the experimentalist who's the first author on the primary paper. So the idea is that you'll submit your models and data to Physiome, and eventually we hope that it'll be at the same time as you submit to your primary journal, so that we can make the models available at the same time as the primary paper comes out. And then if it's successfully peer reviewed by the primary journal, and then we can get it working properly, or we in conjunction with the authors can get it working properly as a SLML model, or any other, whether it's SLML, SPML, NeuroML, doesn't matter, providing it is in a standardized form. Then we can also encourage people to submit review papers that look across the whole area and compare models that are available in these standardized form. And then we've been working with Physiome Science, the publishing group in London, who've been fantastic in helping us to introduce a submission system and templates for Overleaf. So they as part of their portfolio, they've got Overleaf but also Figshare for issuing DOIs, and we're working with Crossref for the referencing. So this is now being driven by Karen, and she can talk a lot more about the detail, but enormous help from the Physiome, from the Digital Science Group in London. And we've been saying this for quite a while, that we're going to have this journal up and going, but I think we seem to be on track for getting it going by the end of this year, where we're aiming to have, we've got 10 papers in development that will be published initially, and then we'll be inviting submissions. And the idea here also was to keep the author, it's got to be open access obviously, but to keep the author processing charge very low. So we're charging $300 per publication, because we figure people are not going to want to pay a $2,000 or $3,000 APC for a second paper, but they would be prepared, most people might be prepared to pay $300 for the Physiome paper. So that means we need to get funding from public good sources to help support the whole, because $300 won't cover the costs of running the journal. But that's where NIH, Wellcome and other funding agencies come in to help. And then the idea is that we'll be creating portals where these models are feeding into larger scale infrastructure that I've talked about before, for a whole range of applications. So go to Karen's poster, or Andre's talk also will talk more about this, and End as well, I think, is a poster on the journal. And part of the idea of the journal is that you can run things directly from the website, click on it and run the models and play with them directly from the website. So I just want to finish by talking a little bit about what I think is the next big challenge for us as well, from a physiology perspective anyway, for community. And that is this issue here, that many models that we see and we can encode in these standards violate the basic laws of physics, especially energy balance. I mean, even Hodgkin-Huxley doesn't satisfy energy balance. So we have to be able to do better than that if we're going to make these models applicable to a wide range of physiological problems where it really matters that you are properly handling energy, not just mass. So the other issue is that a lot of the annotation of these models is done post hoc. You develop your model and then you think, okay, now I want to annotate it to make this variable mean this physically or this biologically. So you bring in all the ontologies to help do that. But it would be far more satisfactory if you had software that could build these models from the ground up, automatically annotating them because you're using well-defined concepts as you build your models, and automatically obeying the laws of physics because you're building them in a way that obeys mass conservation, energy conservation, charge conservation, etc. But the only way we're going to do that is if we go back to basics. Physics should come before maths, is the basic message. The maths encodes the physics, but the key is to get the physical concepts right, and then the maths follows. And it turns out that there is a framework at the lumped parameter level that does this. It's called bond graphs, and it was invented by a guy called Henry Painter at MIT, embarrassingly over 50 years ago, 60 years ago. And the reason he developed it was because he was a professor at MIT who worked in electrical engineering, mechanical engineering and chemical engineering, and he needed to be able to talk between those, to all his colleagues between those departments, and he realized that the only framework, the only thing that's common across all these areas of physics is energy. And of course physiology, any physiological problem involves multiple physics. Every system in the body you need to think about electrical processes, mechanical, biochemical, heat transfer, etc. So physiology is the perfect example of why we must have a framework that properly creates models based on physical principles that obey the conservation laws of physics. And then of course you need all the annotation frameworks. So if we take one step back, all of science in a way starts with units, and there are 7 units in the SI system, but 3 of them really define the world that we live in. They're time meters, so for space, 4-dimensional space-time framework, and energy, joules. So those are the most fundamental units that define the universe that we live in. Then you have another 4 that basically relate to counting the number of fundamental particles involved. So coulombs counts electrons, candela you count photons, moles you're counting atoms, and entropy you're counting probability states. So those are not exactly what are in the SI system, but the SI system about a year ago switched to basically making sure that all its units are now based on these ones. So you no longer have a kilogram defined by a weight sitting in a vault in Paris. You go back to joules and these underlying units in order to define these basic constants. So if you look at mechanics, it involves joules, seconds, meters and entropy. Electrophysiology involves joules, seconds and coulombs and so on. But every physiological system will involve those 7 units. So that should be the starting point, I think. So this is what bond graphs does. Bond graph says you have every process, you have a potential that's the driving, it's providing the energy to drive a process, and then you have a flow, which is the substance being driven, and that substance that quantity will come from one of those basic units. Sometimes they're derived units like bits. You can define in terms of other things entropy, but essentially the basic quantities are the things that are flowing. So current is the flow of coulombs and so on. And then those 2 quantities together, the potential and the flow, their product is power or energy per second, joules per second, and that's the only thing that's in common right across all physical processes. So if you look at mechanics, you shouldn't be thinking about force as newtons. You should think about it as joules per meter, because that's what it is. If you're dealing with electrical systems, don't think about volts, think about joules per coulomb, because that's essentially what a volt is, and so on. Biochemical reactions, joules per mole. Heat flow is the flow of entropy, so you should be thinking about joules per unit entropy, and so on. So the idea of bond graphs is to come back to this basic concept of energy flow, and plus this addition of adding in the basic units to make sure that we're always properly dealing with comparable units across these physical quantities. So you set up a model by defining the topology of a network where you have these things called zero nodes or one nodes. And unfortunately, I haven't got time to go into this in much detail, but basically the zero node is where you satisfy mass conservation. The one node is where you satisfy energy conservation. And then you have, as well as the conservation laws, you have a link to an empirical description, a constitutive relation, which could be a capacitance, for example, if it's a spring for a mechanical system, or a capacitor for an electrical system, or storage of a chemical concentration in a chemical system. In the end, they're all just a capacitance term. Resistance is likewise where you dissipate energy in the system. Inductance, it's either inertia for a mechanical system or electrical inductance for an electrical system. But they're all essentially the same quantity, just expressed in terms of the appropriate units for that discipline. And then the equations pop out automatically. You automatically get the conservation at the zero node for mass and the conservation of energy at the one node, and dealing with storage, dissipation and transmission, and plus the boundary conditions. So we're trying now to bring some of these concepts in to make it easier to construct models based on bond graphs at the lump parameter level through OpenCore. But that's only lump parameter models. The obvious thing is, what about spatial systems? They've also got to be energy based. And it turns out that there is a whole area which hasn't really made its way hardly into physics. It's certainly very strong in the mathematical community, and it's certainly almost unknown in the biological community, but an area called Port Hamiltonians. And Port Hamiltonians essentially does for spatial systems what bond graphs does for lump parameter systems, bases it on energy and makes sure that everything is expressed in a single common framework. And then if you drop from multi-dimensional down to zero-dimensional lump parameter systems, Port Hamiltonians just becomes bond graphs. So it's completely consistent with bond graphs for the lump parameter. And it also has some very nice properties, which I probably haven't got time to go into, but the equations of continuum physics, which really fall into these three categories of either mechanics using the meter, heat flow or reactions using entropy, or electromagnetics from Maxwell's equations for electrical systems. And then I'm going to skip through this very quickly. Essentially, you take the ideas from bond graphs, which are lumped parameter, zero nodes or one nodes, and you extend it to something called a Dirac structure, which is a continuous structure where you maintain orthogonality between these sets of variables that are involved in your power flow. And then you can quickly show, you make use of something called skew symmetric matrices, which allow you to very, very elegantly define these two adjoint variables that you need for power flow. So you end up with systems that look like this, which deal with energy conservation for any area of physics in a completely consistent way, in a way that also nicely separates storage, separates three things: energy coming in from, if you've got a modular system and you've got energy coming in or out into the surrounding environment, from energy storage internally, from energy dissipation. And that becomes critical for, particularly when you're dealing with control problems, because you've got to be able to understand how to move energy around as part of your control system strategy. So Port Hamiltonians are a very elegant mathematical framework for defining these control problems. I'm going to stop there. So SalamL relates to bond graphs as FieldML does to Port Hamiltonians. So what I've talked about is the work of an awful lot of people, a large group in Auckland and with collaborators all around the globe, but particularly for the Physiome project and the current Spark effort, particularly the people here. And I would say particularly Andre, David Nixon, who really is for us the real link with all the standards community in his efforts with Combine. Funding is from a number of New Zealand funding agencies, but also, and from the European Union, but also increasingly now from NIH through these, also the P41 project I mentioned, which Andre's involved in. So thank you.